Right. Okay. Welcome back to our class, biology class. This is now we are going to study about hydrilla plant experiment. That means oxygen liberated in the presence of sunlight. See here, we are going to study about oxygen is liberated in the presence of sunlight during the photosynthesis. So this is the lab activity. So whenever we are going to study about lab activity, we need to follow some steps. That is what first one is aim. The aim of the lab activity is oxygen liberated in the presence of sunlight during the photosynthesis process. Then second one is material. So different material we need, that is beaker, okay, water, then short stemmed funnel, okay, then test tube, okay, incense sticks, etc. So this is about material. Next one, third one is procedure. So what is the procedure? See here, in the procedure, first we have to take one beaker with water. See here, this is the beaker. So in beaker we have poured some water. So 1/3% of water we have poured. Next one, here a short stemmed funnel is to be taken. A short stemmed funnel is to be taken. In this short stemmed funnel, we have taken some hydrilla plants. See here, some hydrilla plants have been taken. These hydrilla plants, why hydrilla plants have been taken? Okay, these hydrilla plants are, see here, hydrilla plants, these are submerged plants. First thing, these are submerged plants. And hydrilla plants absorbs the carbon dioxide from water through epidermis. Generally, carbon dioxide is absorbed into the plant. Carbon dioxide is absorbed into the plant through epidermis because hydrilla plants, see, carbon dioxide is absorbed through epidermis of the plant because here the stomata's number will be reduced. The leaves are reduced or stomatas are reduced. That's why here hydrilla plant is a submerged plant. Then hydrilla plant absorbs the carbon dioxide into the plant through the epidermis. So carbon dioxide already it is in dissolved condition in the water. So from the dissolved condition of from the dissolved carbon dioxide is dissolved now. ఇక్కడ వాటర్లో కార్బన్ డైఆక్సైడ్ డిజాల్వ్ అయింది. ఆ డిజాల్వ్ అయిన కార్బన్ డైఆక్సైడ్ని హైడ్రిల్లా ప్లాంట్స్ ఏం చేస్తున్నాయి? Through epidermis it can absorb. So that's why we are taking hydrilla plant or elodia plant or any submerged plant we can take for this experiment. That plant must be submerged plant. Then this submerged plant it can absorb, especially suppose if you are talking about the hydrilla plant is a submerged plant. It takes the carbon dioxide through epidermis. So that is why we are selected this plant. Next, beaker with some 1/3% of water and short stemmed funnel. This is the funnel. One funnel is taken. Then in the funnel we have put the hydrilla plants. And one more thing, here we have to be taken the test tube. Then this test tube should be filled with some water. This test tube is filled with water. Completely test tube should be filled with water. And this test tube should be inverted over the funnel. How can we invert? First we should put our thumb on the mouth of the test tube, and when we put the thumb on mouth of the test tube, and this test tube should be inverted down to, okay, top to down, itla inverted position lo. So like this, in this manner the test tube should be inverted. See, in the test tube already we filled water, okay, so like this, right? This is test tube. Especially very important, test tube should be inverted position. test tube should be filled with water and test tube should be in the inverted position. This inverted position test tube should be inverted position test tube over the hydrilla plant, which consists of where a funnel. So like this we have to be made some arrangements like this: beaker with some water and short stemmed funnel. In short stemmed funnel, here hydrilla plants have been taken because hydrilla plants are submerged plants. Then hydrilla plants absorbs the carbon dioxide from the water. Carbon dioxide is in dissolved state. This carbon dioxide is absorbed into the plant through epidermis. So like that we have been taken hydrilla plants into the where a short stemmed funnel. Then over the short stemmed funnel, here one test tube is to be taken. Test tube must be filled with some water. This test tube filled water should be inverted over the funnel by keeping our thumb. Like this we have to be made some arrangements. So like these two arrangements we have to be taken. One arrangement should be kept in the sunlight, one arrangement should be kept in the dark room. If we observe after few hours or after 2 to 3 days, maximum after 2 to 3 days or after few hours, if we can observe what happens, from these hydrilla plants we can observe some air bubbles coming out. Some air bubbles coming out through the hydrilla plant funnel. ఇక్కడ ఫనల్ ఉంది కదా, ఈ ఫనల్లో ఉన్న హైడ్రిల్లా ప్లాంట్స్ నుండి మనకు కొన్ని ఎయిర్ బబుల్స్ అనేవి ఇలా బయటకు వస్తూ ఉంటాయి. ఏంటి మరి ఎయిర్ బబుల్స్? దీస్ ఎయిర్ బబుల్స్ ఆర్ ఫైనల్లీ రీచింగ్ టు ద టాప్ ఆఫ్ ది టెస్ట్ ట్యూబ్ అండ్ దే వర్ స్టోరేజ్డ్ దేర్. This gas is storaged at the top of the test tube. So now we have to be identified what type of gas it is. ఈ హైడ్రిల్లా ప్లాంట్ ఫనల్లో నుంచి హైడ్రిల్లా ప్లాంట్స్లో నుంచి వచ్చే ఈ గ్యాస్ మరి ఏంటి అని మనం తెలియాలంటే ఏం చేయాలా? జస్ట్ take out the test tube from the water by putting our thumb on the mouth. Same thing, just you can put our thumb on mouth of the test tube and remove the test tube from the water. Suppose if you remove the test tube from the water up to water level, no gas can be leakaged, but when the test tube is open out above the water, then the gas will be sent out. That's why we should put our thumb at the mouth of the test tube. When the test tube comes out of the water, put the thumb and you should be the test tube should be kept in the straight position and insert one incense stick. If you put one incense stick into the test tube, ఇలా టెస్ట్ ట్యూబ్ తీసి మన థంబ్ ఇలా టెస్ట్ ట్యూబ్ మౌత్ దగ్గర పెట్టి దాన్ని జాగ్రత్తగా తీసి స్ట్రైట్గా పెట్టేసి మనం ఒక ఇన్సెన్స్ స్టిక్స్ని తీసుకొచ్చి నెమ్మదిగా ఈ థంబ్ ఓపెన్ చేసి దాంట్లో పెట్టేసినాం అనుకో, ఇఫ్ ద ఇన్సెన్స్ స్టిక్ బర్న్స్ వెరీ బ్రైట్లీ, that shows about oxygen gas. That means oxygen encourages the burning. So with this we can understand, in the presence of sunlight oxygen is liberated. In the presence of sunlight these hydrilla plants perform the photosynthesis. When the photosynthesis occurs, the sunlight energy is utilized. At the same time carbohydrates are formed. At the end of the photosynthesis oxygen gas is liberated. So that oxygen gas liberated in the presence of sunlight. So this is procedure, observation, conclusion, and finally precautions. We have to know the precautions also. What precautions? Test tube must be in the inverted position. Okay, here test tube should be kept in the inverted position, and when we keep the test tube in inverted position, before keeping the test tube should be filled with water. Otherwise, if empty test tube is inverted onto the funnel here, if the empty test tube is inverted onto the funnel, what happens? Empty test tube means there might be some air, so that we cannot identify whether the gas is released from the hydrilla plant or the air. So that test tube should be filled with water, then after the test tube should be inverted on funnel, so that we can identify the gas. And one more important thing, in this experiment only submerged plants must be taken, that is like hydrilla plants or elodia or any submerged plant should be taken for the experiment. And to know the gas, here we must introduce what incense sticks or burning any match stick should be inserted so that it can burn very brightly. That's why it is known that it is oxygen gas. So with this we can understand oxygen liberated in the presence of sunlight, so that sunlight energy is utilized in the photosynthesis and carbohydrates are formed. Oxygen is liberated. But the same experiment when it is done in the dark room, no water, from the water no air bubbles are produced, so that we can understand sunlight energy, it is absent in the dark room. When the sunlight energy is absent in the dark room, hydrogen plants cannot perform the photosynthesis. No formation of carbohydrates or no producing the oxygen, no liberation of oxygen. That's why we can understand oxygen liberated only in the presence of sunlight. So that sunlight energy is essential for photosynthesis.